Chapter 9, The Organ Tree. I have yet to see any problem, however complicated, which when you looked at it the right way did not become still more complicated. Science fiction writer Paul Anderson once observed, "To a certain extent, this is true of regeneration. Intricate nature is still more than a match for our finest spun hypothesis. Yet we've now reached the oasis of science that we call an interim understanding, where the data begin to shake into place and we can sense the pattern of this robust from the blanks we've filled in. Ultimately, we must relate all we learn about regeneration to a general system of communication among cells. For regrowth is only a special case of the cooperative cohesion that's the essence of multicellular life. This communication system includes, but extends beyond the gene protein enzyme subsystems that govern the specialization of cells and unite their chemical trade routes into smoothly working tissues and organs. During embryonic development, cells where muscles will appear must receive instructions from their environment, telling them to repress all genes except the muscle genome or subcode. In many tissues, perhaps in all, chemical inductors from previous formed tissues perform this task, leading embryonic cells step by step through the stages of differentiation. However, chemical reactions and the passage of compounds from cell to cell can't account for structure, such as the alignment of muscle fiber bundles, the proper shape of the whole muscle, and its precise attachment to bones. Molecular dynamics, the simple gradients of diffusion, can't explain anatomy. The control system we're seeking unites all levels of organization, from the idiosyncratic yet regular outline of the whole organism to the precisely engineered traceries of its microstructure. The DNA RNA apparatus isn't the whole secret of life, but a sort of computer program by which the real secret, a control system, expresses its pattern in terms of living cells. This pattern is part of what many people mean by the soul, which so many philosophies have tried to explicate. However, most of the proposed answers haven't been connected with the physical world of biology in a way that offered a toehold for an experiment. Like many attempts, the latest major scientific guess, the morphogenic field proposed by Paul Weiss in 1939, was just a restatement of the problem through a useful one. Weiss conjectured that development was guided by some sort of field projected from a fertilized egg as the dividing cell mass became an embryo and then an adult. The field changed its shape and somehow led the cells onward. The problem was that there were too many somehows. Even if one accepted Burr's largely ignored measurements of an electric L field and admitted that it might be the morphogenic field, a possibility Weiss dogmatically rejected, there was still no way of telling where the L field came from or how it acted upon cells. Nor was there an explanation of how, if the field was an emanation from the cells, it could also guide them in building an animal or plant. In applying the idea to regeneration, biologists face the related and seemingly insurmountable problem of how a more or less uniform outflow of energy could carry enough information to characterize a limb or organ. Given the complexity of biological structures, this was even harder than imagining how a field could somehow survive when the part it referred to was missing. However, the morphogenic field no longer has to account for everything. Acceptance of dedifferentiation lets us divide regrowth into two phases and better understand each. The first phase begins with the cleanup of wound debris by phagocytes, the scavenger race of white blood cells, and culminates in dedifferentiation of tissue to form a blastema. Redifferentiation and orderly growth of the needed part constitute the second phase. Simplifying the problem in this way should give biologists an immediate sense of accomplishment, for the first stage is now well understood. After phagocytosis, while the other tissues are dying back a short distance behind the amputation line, the epidermal cells divide and migrate over the end of the stump. Then, as this epidermis thickens into an apical cap, nerve fibers grow outward and subdivide to form individual synapses-like connections, the neuroepidermal junction, NEJ, with the cap cells. The connection transmits or generates a simple but highly specific electrical signal in regenerating animals. A few hundred nanoamperes of direct current, initially positive, then changing in the course of a few days to negative. The pituitary hormone prolactin, the same substance that stimulates milk flow in nursing mothers, seems to sensitize cells to electric current. Then the signal causes nearby cells to dedifferentiate and form a blastema, apparently by changing the way cell membranes pass calcium ions. After confirming our frog blood cell work, Art Pilla went on to produce the same changes by using pulse DC to make a wave of calcium ions flowing across the culture dish. Steve Smith then confirmed the importance of calcium by preventing dedifferentiation with a calcium blocking compound and restarting it with another substance that enhanced passage of calcium ions. Working together, Smith and Pilla next used the same PIMP wave form now in clinical use to nearly double the rate of salamander limb regeneration, while completely preventing it with a different pulse pattern. Widespread recent work on calcium binding proteins such as calmodulin, has made it fairly certain that electrical control of calcium movement through cell membranes direct the give and take among these proteins, which in turn supervises the cell's entire genetic and metabolic industry. Although not conclusive, the available evidence suggests that the current flows through the perineural cells rather than the neurons themselves. These are several types of cells that completely surround every nerve cell, enclosing all the peripheral fibers in a sheath and composing 90% of the brain. Lizards can replace their tails without the spinal cord as long as the epiderma, or perineural cells, surround the cord remains intact, and epidermal tissue transplanted to leg stumps give lizards some artificial regeneration there. However, the circuit may shift tissues near the wound, for Elizabeth Hayes' electron microscope studies clearly show that the peripheral nerve Schwann cell sheath stops just north of the epidermis, and only the naked neuron tips participate in the NEJ. The exact current pathway is this microscopic area remains to be charted. Not all cells can respond, however, as Jim Cullen and I found in one part of our fortuitous rat regeneration experiments of 1979. Dedifferentiation occurred only when we passed the deviated sciatic nerve to the epidermis through the bone marrow. When we led it through the muscle, yet sutured it to the skin in exactly the same way, an NEJ produced the right current appeared, but no blastema and no regrowth. Muscle cells apparently weren't competent to dedifferentiate in the adult rat. The cellular target proved to be just as important as the electrical arrow. As the electrical arrow, sorry. There's still some opposition to parts of this scenario. Among some scientists, the prejudice against electrobiology remains so strong that one otherwise fine recent review doesn't even mention the NEJ or the difference between currents of injury in frogs versus salamanders. Other objections are a little more substantial. A Purdue University group has measured electrical potentials near the surface of regeneration limbs underwater using a vibrating probe. This is an electrode whose tip, ending in tiny platinum ball, oscillates rapidly to and fro, giving the average voltage between the two ends of its motion. The researchers describe an arc of ion flow. They categorically deny the possibility of electron currents in living tissues. Out from the stump and through the water, or in semi-aquatic animals, a film of moisture on the skin. From the water, they suggest these ions travel to the limb skin behind the amputation, then and through all the inner tissues, and finally out the stump again to complete the circuit. They believe the epidermis drives these currents by its normal amphibian function on pumping sodium ions from the outside water into the body. They conceive of this ion flow as the regeneration current itself because changing the concentration of sodium in the water directly affects the current measurements, and because certain sodium blocking techniques have interfered with limb regrowth in about half of their experimental animals. Of course, the Purdue researchers don't dispute the proven need for nerve and injury currents during regeneration. They've even confirmed Smith's induction of leg regrowth in frogs with batteries generating electron currents. Nevertheless, they consider nerves target rather than the source of current, even though they propose no reason why their ion flow should be restricted to nerve tissue. In fact, they base their hypothesis partly on evidence that sodium flows even from degenerate denervate limbs. There are several other problems with this theory. Its proponents' own measurements show that the sodium ion current almost disappears just when its supposed effect, blastema formation, is occurring. Moreover, it fails to explain the easily observed reversal of polarity in injury currents measured directly on the limb, as well as the crucial role of the NEJ. The proposed circuit goes right past the NEJ. Finally, it can account for the several tests of semiconducting current throughout the nervous system or regeneration in dry skinned animals such as lizards. To the layperson, all this may seem like academic hair splitting until we reflect on the stakes, understanding regeneration well enough to restore it ourselves. Certainly, skin is electrically active. It's piezoelectric and pyroelectric, turning heat into electricity, as well as the transporter of ions in wet animals. In the last 2 decades, nearly all tissues have been proven to produce or carry various kinds of electrical charge. Skin may play as yet unknown role in regeneration besides its part in the NEJ, or it may merely be producing unrelated electrical effects. In any case, there are far too many data about the role of nerves to call skin the major source of the regeneration current. In fact, even the Purdue group has measured a step current that's independent of sodium concentration. A recent experiment by Merrill Rose gave further evidence of neural DC without clearing up all aspects of the question. Rose removed the nerves from the larval salamander legs before amputation. Normally, such denervated larval stumps die right back into the body wall, but when Rose artificially supplied direct currents like those I measured in my first experiment, they regrew normally. This is pretty conclusive proof that the nerves are the electrical source in phase one. However, since it looks as though nerves are organized regeneration's second phase, see below, it's hard to understand how the new legs could have been completely normal when they were disconnected from the rest of the nervous system. Perhaps salamanders can pinch hit for nerves at the stage through a tissue other than the nerve. On the other hand, new nerves may simply have regrown into limbs and be noticed to Rose by the later stages of the experiment. Phase 2 begins as the embryonic cells pile up and the blastema elongates. Early in this stage, a sort of spatial memory becomes fixed in the blastema cells so the limb to be will have its proper orientation to the rest of the body. At the same time, or shortly afterward, the cells at the inner edge of the blastema receive their new marching orders and platoon assignments. They redifferentiate and take their places in the new structure. We can infer two things about the control for this part of the process. Since a blastema forms the right structure in relation to the whole organism, the guidance can't be purely local, but must come from a system that likewise pervades the whole body. Furthermore, there are no dedifferentiated cells left over when the work is done. There are just enough and no more. Thus, there must be a feedback mechanism between the redifferentiation controls at the body side of the blastema and the NEJ dedifferentiation stimulus at its outer edge. A large body of earlier work has shown that the redifferentiation instructions are passed along a tissue arc, whose main element is the circuit already established between nerves and epidermis in the first phase. The electrical component persuasively explains how this arc and update of the morphogenic field may work. The direction polarity plus the magnitude and force, amperage and voltage of current could serve as a vector system giving distinct values for every area of the body. The electric field surrounding continuously charged cells and diminishing with the distance from the nerve would provide a third coordinate, giving each cell slightly different electrical component. In addition, a magnetic field must exist around the current flow, possibly adding a fourth dimension to the system. Together, these values might suffice to pinpoint any cell in the body. The electric and magnetic field varying as the current varies with the animal state of consciousness and health could move charged molecules wherever they were or needed for control of growth or other processes. Since currents in electromagnetic fields affect the cell membrane's choice of what ions to absorb, reject, or expel, the system, in concert with the chemical code by which neighboring cells recognize each other, could precisely regulate the activities of every cell. It could express the exact point along the limb at which new growth must start, distinguished between right and left, top and bottom, even explain how totally missed parts like extirpated bones or all of the little bones of wrist and hand can reappear. Furthermore, the difference between electrical values at the inner and outer edges of the blastema would lessen as a new limb grew behind it. Remember that the electrical potential grows increasingly negative the end of the intact limb. The gradual convergence of these two values could constitute a feedback signal perfectly reflecting the number of dedifferentiated cells still needed. Although the results weren't entirely conclusive, perhaps because measurements had to be made under anesthesia, several experiments in the 1950s suggest that such voltage differential governed restitution of the proper numbers of segments in earthworms. There was even a surge of positive potential that seemed to indicate when the job was finished. This is a rich concept, and the details are without doubt more complex than this sketch, but they're all open to experimentation in a way that Weiss's morphogenic field and Burr's L field were not. The best part of this two-stage analysis is that it gives us a rationale for trying to foster regeneration after human injuries before we know all the details of the second phase. The rat limb experiments strongly suggest that mammals lack two crucial requirements for the first phase of regeneration. They don't have the necessary ratio of nerve tissue to total limb tissue, the amount needed to make the dedifferentiation stimulus strong enough, and they lack the sufficient sensitive cells to respond to the electrical stimulus and form a big enough blastema. The work on rats pointed the way to defining the proper current, and the ability of electrical injected silver ions to dedifferentiate fibroblasts now gives us a positive method for producing an adequate blastema. We should now be able to supply the requirements for phase 1 in humans. Once this is done, the body itself can probably take care of phase 2, even though we don't understand the process. Fingertip regrowth in children suggests that our bodies still have the ability to redifferentiate cells and organize the missing part as long as the electrical stimulus and the supply of sensitive cells are sufficient. Microsurgeons have performed wonders in reimplanting cleanly in severed portions of arms, legs, and fingers, but these limbs are subject to atrophy and obviously can't be grafted if they're too badly mangled or riddled with disease. As one who has performed too many amputations in this time, I find the prospect of being able to give a patient the real thing instead of prosthesis tremendously exciting. There's a good chance that we'll eventually treat some non-genetic birth defects or old injuries by cutting off the defective part and inducing a normal one to grow. Perhaps combined with gene splicing, such techniques could even rectify genetic birth defects. Since no one has yet achieved full regeneration in rats or any other mammal, these dreams won't come true overnight. They aren't chimerical, however. The remaining problems could probably be solved in a decade or two of concerted basic research. Meanwhile, human capacities for repair of certain tissues are greater than most people realize, and there are already promising ways of enhancing some of them. Cartilage. Fossils show that even the dinosaurs had arthritis, but unfortunately it outlived them. Many varieties have been described, all of which in destruction of the hyaline glassy cartilage that lines the ends of the bones. The remaining cartilage cells try to heal the defect of proliferating and making more cartilage. They're almost never equal to the task, and scar tissue fills the rest of the hole. The result is pain, for scar tissue is too spongy to bear much weight or keep the bones from grinding against each other. After our success at getting rat legs to partially regenerate, we studied this problem in 1973. We reasoned that since cartilage was made by only one kind of cell, getting it to regrow would be easier than working with a whole limb. With orthopedic resident surgeon Bruce Baker of Upstate Medical Center, we removed the cartilage layer from one side of the femur at the knee in a series of white rabbits. The operation left a circular hole of the bare bone about 4 millimeters across. In the experimental animals, we implanted silver platinum couplings like those used on the rats, drilling the platinum end into the defect and tucking the rest along the bone. Most of the control animals filled in the defect with scar tissue along with some of inferior fibrous cartilage. About a tenth of them grew a millimeter or two of good hyaline cartilage at the edge of the hole. But sure enough, the rabbits with the implants showed greatly enhanced repair. When we used an improved battery implant with silver wires at each end, we got an even better result. Two of the rabbits healed the damage completely with beautiful hyaline cartilage, just like their original material. A few years later, when we were testing various electrode metals, we tried a different approach specifically for rheumatoid arthritis, in which runaway inflammation causes phagocytes to attack healthy cartilage cells. Gold salts taken orally sometimes control this disease, but often produce toxic side effects. We figured electrical injection of pure gold directly into the joint with no other ions might work better. To find out, Joe Spadaro and I produced rheumatoid arthritis in the knees of both hind legs in 40 rabbits with using a standard experimental procedure. Then we treated one knee in each animal with a positive gold electrode stuck right into the space between the two bones for 2 hours. Joe did the actual treatments. Then we sacrificed the animals gradually over a period of 2 months, and I examined both arthritic knees, not knowing until latter which had been given gold. During the first 2 weeks, about 70% of the treated knees were markedly better than the untreated ones. The improvement fell off to about 40% thereafter, suggesting that the treatment must be repeated for continued results. Obviously, these were only preliminary experiments. However, since an estimated 31 million Americans suffer from arthritis, for which there is no cure yet, I think both avenues should be thoroughly explored as soon as possible. Skull bones. Lev Polichev has spent his career investigating what might be called the Polichev principle. The greater the damage, the better the regrowth. He has found he can often enhance repair by adding homage homogenates, minces, and extracts of the damaged organs, even though this doesn't augment the current of injury as his needling procedure did. Eventually, Polachev developed a way to induce regeneration of holes in the skull, which normally heal over with scar tissue. As long as dura mater, the tough membrane between skull and brain, is intact, a paste of blood and fresh, living powdered bone will induce the bone cells at the edge to grow and bridge the gap. Microscopic studies have shown that the few live cells remaining in the paste don't survive and the bone particles themselves soon dissolve. Instead, some substance from the disintegrated bone stimulates repair. Since this is first successful trial in humans by several Russian surgeons in the mid-1960s, this method has gradually come into increasing use in the Soviet Union. There is at present no indication whatsoever that humans could ever regenerate any part of their eyes, but the ability of newts, salamanders of the genus Tritarius, to do so makes this tantalizing research ideal for the far future. If the lens in a newt's eye is destroyed, the colored cells of the top half of this iris extrude their pigment granules, then transform by direct metaplasia into lens cells. They soon start synthesizing the clear fibers of which the lens is made, and the whole job is finished in about 40 days. In the case the iris is gone too, a newt can create a new one from cells of the pigmented retina, and those cells can also transform into neural retina layer in front of them. If the optic nerve gets damaged, the neural retina in turn can regenerate the nerve tract backward and reconnect it properly with the brain. No one knows why newts are so much more adept at this than all of the other creatures. Their eyes have no obvious structural or biochemical peculiarities. Steve Smith gave us an important fact to work with when he found two proteins in the lens that seem to prevent the iris cells from changing into new lens cells as long as the old lens is in place. Since the neural retina must be intact for most of the transformations to happen, it may provide a constant electrical stimulus that goes into effect only when the inhibitory proteins are removed by injury. No blastema is formed. Instead, the cells change costume right on stage. Furthermore, certain ingenious experiments have shown that a wound isn't really necessary, only the interruption of the inhibitory mechanism. Therefore, the stimulus from the neural retina probably isn't the familiar injury current of limb regrowth. However, despite a voluminous research literature on newt eye regeneration, no one has yet studied its electrical aspects. This may be why we're still so far from understanding the natural process, let alone trying to adapt it to human eyes. Muscle. Every muscle fiber is a long tube filled with rows of cells, myocytes, laid end to end with no membranes between them. In effect, one nucleated cell called a syncytium. These nuclei direct the manufacture of contractile proteins, which are lined up side by side and visible when stained as dark bands across the array of myocytes. Each muscle fiber is surrounded by a sheath, and groups of them are bound together in bundles by thicker sheaths. At the edge of each bundle are long cylindrical cells with huge nuclei and very little cytoplasm, called myoblasts or spindle cells. Also, along the edges between the spindle cells, clusters of very tiny satellite cells can be seen at high magnifications. After a crushing injury or loss of blood from a deep cut, muscle in the damaged area degenerates. The myocyte nuclei shriveled up and the cells die. Soon phagocytes enter to eat the old fibers and cell remnants. Only the empty sheath and few spindle and satellite cells are left. Now these remaining cells turn into new myocytes, fill up the empty root tubes, and begin secreting new contractile proteins. Although the early part of this process proceeds without nerves, it can run to completion only if motor nerve fibers reestablish contact with the terminals, called end plates, that remain at specific distances along each fiber sheath. If these end plate areas are cut out, the nerve endings will enter, sniff around, and then retract. The muscle will atrophy. The nerve reestablishes the connections. New muscle cells will fill most of the original volume, gradually build up strength, and then completely differentiate into slow twitch or fast twitch fibers. Attempts to enhance muscle regeneration in humans take two approaches, if available. A graft of a whole muscle from another source is more effective. This is actual single tissue regeneration because the original muscle cells die and are replaced after new blood and nerve connections are made. Since its first clinical use in 1971, this approach has proven successful in replacing defective small muscles of the face and also is restoring anal sphincter control. Large limb muscles haven't been successfully transplanted yet. Another method may soon be used in humans when grafts aren't possible. Muscle regeneration in birds and laboratory animals has been considerably enhanced by inserting muscle tissue minced with fine scissors into pieces of no more than 1 cubic millimeters. Soviet biologist A.N. Studinsky first devised this method in the 1950s, extending Polichev's work, but its development has been slow. Abdominal organs. Despite over 200 years of descriptive work, new regeneration capacities are still discovered in animal kingdom from time to time. We recently learned, for example, that adult frogs can restore their bile ducts, although for some reason females are better at it than males. Doctors have long known that the liver can replace most of the mass lost through injury by compensatory hypertrophy, in which cells both enlarge and increase their rate of division so that the organ's chemical processing duties can be maintained even though the ruined architecture isn't restored. Similarly, damaged in one kidney is made up by enlargement of the other without rebuilding the intricate mazes of microtubules in the glomeruli. Recent studies of rat liver suggest that a combination of insulin and epidermal growth factor, modified by at least a dozen other hormones, enzymes, and food metabolites, control the cell proliferation. Now it appears that the spleen can make the same kind of comeback, at least in children. Adults who must have their spleens removed rarely miss them, but children become more susceptible to meningitis. A few years ago, medical researchers noted that children whose excised spleens have been damaged by the accident were less likely to get meningitis than whose spleens had been removed because of this disease. Howard Pearson and his colleagues at the Yale University School of Medicine found that when ruptured, the delicate spleen left bits of itself scattered in the abdomen, which grew and gradually resumed the organ's obscure blood cleanse function. Now, when they remove a spleen, many surgeons wipe it on the peritoneum, the tough membrane that lines the abdominal cavity, to sow replacement seeds. Another late discovery of regeneration was made in the late 1950s when several scientists learned that tadpoles, larval salamanders, and sometimes adult salamanders could restore up to about 4 inches of their intestines. Moreover, all adult amphibians could reconnect the cut ends if they couldn't replace a missing section. Alan Dumont, one of my best friends during medical school, and now Jules Leonard Whitehill, professor of surgery at NYU Bellevue, decided to check this potential in mammals after I told him about my work on rat limbs. He wanted to find out whether regeneration could be stimulated in mammals to solve one of the general's surgeon's most vexing problems: poor healing of sutured ends of gut after a cancerous or degenerated segment has been cut out. Even a small opening can spill feces into the abdominal cavity, with disastrous peritonitis result. Like any good scientist, Al started from the basics. After several years, he confirmed the earlier reports. When he cut pieces from the gut of adult frogs and newts and merely put the ends together in the abdomen, 40% of the animals survived by quickly reconnecting the two ends and completely healing them in about a month. Although the newts didn't replace much of the lost length in this experiments. Gut regeneration actually involves several tissues. AL's cell studies showed a blastema quickly forming at the junction and then differentiating into smooth muscle, mucus cells, and the structure cells of the villi. Naturally, when I was organizing a conference on regeneration in 1979, I invited Al to present his results. About a month before the meeting, after the program had already gone to the printer, he wanted to change the title of his paper. For he'd just finished some surprising work, he'd asked me, "What would you expect to happen if I took some adult rats, cut out a centimeter of gut, and dropped the two loose ends back into the abdomen?" Like any first year med student, I said they'd be dead of periodontitis in 2 or 3 days. Well, 20% of Al's rats had reconnected their bowels better than surgery could have done and were alive and healthy. When Al had given one group of animals a temporary colostomy above the experimental cut, the survival rate jumped threefold. The perfect healing of the test animals compared with the controls indicated that sutures actually interfered with the regeneration, producing unnecessary scars and adhesions. No one knows for sure how the two cut ends find each other, but there's certainly some active search going on. For periodontitis, for periodontitis sets in too quickly for the results to be due to chance. The process resembles a regrowing nerve fiber search for its severed part, which may be conducted by electrical factors, a chemical recognition system, or both. Electrical potentials probably play the most important part, for recent research has found DC potentials at injuries on this peritoneum, and experimental changes in the peritoneum's normal bioelectric pattern attract the inner membrane and closing the bowels, closing it to adhere to the side of the disturbance.